Welcome to BioMars. Hello everyone. Today I'm gonna talk about epigenetics. To me, epigenetics is the most interesting part of biology, and currently I'm internship in the lab, and the lab is talks research in the epigenetics. So why I choose epigenetics as today's topics? Because I've read a lot of literature and watching a lot of videos about epigenetics to understand my lab project. However, I think their explanation are very hard, very confused. Even I read like about like 5 or 6 papers and numerous videos. I still cannot understand epigenetics. So today I gonna use very short and clean sentence to introduce talks about epigenetics. So the first is what is epigenetics? Epigenetics basically is two things, but it only have one aim. The only aim is change phenotype without change genotype. As we mentioned on the evolution video, we know the phenotype and the difference between the genotype. Genotype basically is a gene or some molecules, some cells. And phenotype is what we act like. For example, somebody are tall, somebody are short, somebody are um more lighter, have a lighter skin, and somebody have the like darker skin, so they are phenotype, which is kind of visible, and genotype is invisible, is intercell or intermolecular. So how we can change the phenotype without change any genotype? It's really hard, right? Because if we want to change the phenotype, we have to change the gene. Like for example, a darker skin parents prefer to have a darker skin child because they have a such a darker skin gene. And um for lighter skin, parents prefer to have a lighter skin child because they have a lighter skin gene. So how can we do this? Like without, because we can active or silence gene expression by using the histones. Like we all know the histones is on the DNA, and we can regulate the histone to active some genes or silence some genes. For example, still a darker skin parents, we can just silence their darker skin gene to make their maybe their next generation skin become more lighter. It's just an example to understand better. So I'm not sure do we have darker skin gene, but it will be very easy to understand example. So second question is, what is histone? Histone is a protein that provides structural support for a chromosome. So basically, it's support DNA because DNA built up by chromosomes. And eukaryotic DNA is packaged and wrapped around proteins known as histone, which protect and regulate gene expression, just like I mentioned above. The structure of DNA wrapped around histone is known as chromatin. Chromatin is the first level of the organization, appears as a linear array of uniform structure units, nucleosomes. So, and also the both DNA and histone proteins are preferred to methylation, and is associated with only about the histones. So the modification governs the gene expression pattern in a cell by alter between transcriptional activation and repression. So that's how the histone can change the expression of DNA. It can active or repress. We also can cause silence gene expression. So that's how we can control the epigenetics, or we can control the expression of genes. But the histone moving. History is kind of moving, right? It's very complicated. So it's also a really hard topic